Hello, welcome to another session of Bioinformatics. I am Dr. Y.V. Rajesh. Today we'll discuss about sequence comparisons, local and global pairwise alignments. So as a part of Bioinformatics, we are discussing various topics related to biology as well as informatics. Previous sessions we learnt about the actual biological information and how the information is stored in databases and how can we download or access that information. So from today's session onwards, we'll do something called as sequence alignment, which forms the heart of bioinformatics. So we'll learn about pairwise sequence alignments or pairwise sequence comparisons followed by multiple sequence alignment. So our topic is sequence comparisons, local and global pairwise alignments. So why sequence comparison is done? As I told you, it is heart of bioinformatics, and this is the first step towards structural, functional and phylogenetic analysis of a newly determined sequence. So it is a first step in bioinformatics here. So when you have an unknown protein which you got through sequencing technologies, so if you want to draw some evolutionary or functional inference for the unknown protein or unknown DNA sequence, the first thing you are going to do is sequence alignment. So to draw functional and evolutionary inference, so we'll do comparisons here. So why to align sequences? So sequence alignment, you have to take it as two kinds of problems. One is biological problem and second one is computer science problem here. So why it is a biological problem? Because to know the function and structure of the sequence in one dimensional as well as when it is forming to three dimensional protein here. And it is a computation problem because sequence alignment is comparisons of characters. You can call it as a natural language processing problem to find out where the exact words are matching or where the optimally aligned words or characters are matched here. So sequence alignment, it has functional significance to discover the functional aspects of unknown protein or unknown sequence, and structural aspects if it is a protein, and evolutionary relationships among different genomes or different organisms here. So by using simple comparisons, you have two or more genes, you can construct a evolutionary history and also tell the functional and structural similarity here. So while doing sequence alignments, so the first step is doing pairwise sequence alignment. So pairwise sequence alignment, as the name says, a pair of sequences, two sequences of DNA or two sequences of protein, or you can take a DNA sequence and protein sequence and you can compare by translating DNA to protein or by reverse translating protein to DNA. So you require two sequences here. So when you are aligning the sequences, there are so many ways you can do the alignments here. So lot of possible alignments, so which is a computer science problem, and mathematically any two sequences can be aligned, and while doing the alignment, so mathematically you can provide the score here. So basically you can align two sequences here, and evolutionary basis is there for the sequence comparisons. So DNA and proteins are considered as products of evolution. So if you understand the evolutionary history of our planet Earth, so all living organisms came from a common ancestor. We call it as last universal common ancestor. So from this one you got archaea, eukarya and bacteria, and one is from a single preliminary species or primordial species, complex human being came here, and the commonality between the preliminary species, our ancestor, and a human being is the DNA and protein is present, and the part of DNA or part of protein may be still similar after billions of years of evolution. So we can consider DNA and proteins are products of evolution, and they are also considered as fossils that have millions of history of evolution. So during the inheritance or during the time progresses, during the evolutionary processes, the DNA and protein undergo random changes, and some of them may undergo mutations or changes, and some of them may not change at all, may not change, or they slightly change here. So we can consider the characters or the residues which are not changed. You call them as conserved characters or conserved positions, and you can trace the evolutionary history. And the presence of evolutionary traces is because of key functions and structural roles. For example, if you see the RNA polymerase in E. coli, and in broader sense, the transcription machinery in E. coli and in yeast or human being, they are conserved here. The sigma protein actually is similar to TF2D protein of human being here. So that means evolutionary traces are there because both perform similar functions here. And while doing the sequence comparisons, so you have to do what you are going to compare, what you are going to do comparisons here. So we know that DNA is made up of 4 different kinds of nucleotides, A, T, C and G, whereas protein is made up of 20 amino acids. So when you see proteins has 20 amino acids, a lot of variety compared to DNA. So when you are comparing two sequence of DNA, if match is present, that may happen because of randomness. Randomly also matches can happen here. But while you are comparing proteins here, if matches are present, that is purely because of non-randomness nature or conservative nature of the amino acid residues. So while doing comparisons here, so if you have protein, you go for the protein sequence comparisons. And another important aspect is, so if you see the proteins in 20 amino acids, several classifications are there. You have tiny amino acids, small amino acids, positively charged amino acids, polar amino acids, charged, hydrophobic, aliphatic, aromatic. Different kinds of classification of amino acids are there, 20 amino acids. You already know, learned in your biochemistry courses previously here, right? So here, if evolutionary conserved, if your position is evolutionary conserved, it is allowed to change to an amino acid of similar structure and function. Suppose you have aliphatic, isoleucine, leucine and valine. If mutation happens from isoleucine to leucine, so you can say that this is a conserved position here. Conserved position. Suppose if isoleucine is becoming to tryptophan, you can say this position may not be conserved here. So this is a gross definitions I am saying here. So proteins have lot of variety. So they also have different kinds of classes, chemical similarity and functional similarity of amino acids are there. So that's why we go for protein comparisons here. And so when you are doing comparisons, so you say comparisons in the context of 3 things: identity, similarity. 3 things: identity, similarity and homology. 3 terminologies we broadly use it. So identical. So when you take 2 characters, suppose I am taking 2 protein sequences, 2 random protein sequences. So I will say valine, lysine, arginine and serine, and then next sequence I am taking valine, lysine, serine and threonine. So I am taking, so in this one the first position is same in both, second position is same in both. So you can say these are 50% they are identical. 50% identical characters are present in these 2 sequences. So now similarity. So since it is a protein sequence, so similarity context can be applied, can be extended to these 2 sequences. If you see serine is a OH containing amino acid and threonine is also OH containing amino acid. So these 2 amino acids are similar in nature, and so you can consider as a 75% similarity. So 50% identity and 75% identity, 75% similarity is there. So identity and similarity are quantitative statements. You tell in terms of numbers, and there is something called as homology, which is a qualitative statement. You say yes or no. So while doing sequence comparisons, so you need to consider the percentage of matching which is present between amongst the sequences here. So for DNA sequences, generally if you have 70% 70% similarity, then you can consider that these two sequences may be evolutionarily have similarity, evolutionarily, or you can say evolutionarily they are significant, their comparisons here. So but while coming to the proteins, your cutoff can start from 25%. As I told you, 25%, maybe value may look smaller here because lot of variety is there in the proteins here, and functional sites may be very much important for proteins, and remaining sites can undergo lot of mutations here. So you start from the 25% for the proteins here. So here is a graph showing the sequence identity and sequence length for protein comparisons, and you can divide three kinds of zones based upon our identity here. So midnight zone, twilight zone, and safe zone. So midnight zone means we cannot consider that one, consider the percentage of similarity as a evolutionarily or statistically significant similarity. So less than 20%, whatever the length, so x-axis is the length of the sequence, whatever the length you cannot consider as a significant here. So if your length is increasing here, so somewhere here the two lines are matching, so here sequence identity, when longer sequences are there, you can consider less identical sequences, and short sequences are there, you have to consider higher identity here. So twilight zone, so beyond 25%, beyond 20 to 25% is called as twilight zone, and extending beyond 30% or even beyond 30% for many of the sequences, we call it as a safe zone here. So you remember one point here, for DNA 25% and proteins 25% and DNA 75%, identity should be there while doing the sequence comparisons. So we learned about identity and similarity. So we'll learn about, there is a qualitative statements called as homologs. So homologs, orthologs, paralogs, and genealogs, four terminologies we use while comparing the sequences in qualitative approach. The definition of homologs is something like